That already brings us to the last section of this webinar. We'll be talking about the next steps towards implementation of DNA-based methods, and this will be presented by the GEANS coordinator, Annelies De Backer from ILVO, and will also include what was supposed to be 2 pitch presentations, but I understand that one of the presenters has bad connection and couldn't call in, so it will be 1 pitch presentation on Horizon Europe project Obama Next by Peter Steyer from Aarhus University. And then I would suggest that the other project pitch, which was about the Marco Bolo project by Matthias Obst, will be put online on the website, and you can of course contact Matthias with any questions you have later. So with that, I will give the floor to Annelies for our final presentation.
Yes, good afternoon, everyone. Thank you, Louise. As you've just seen in the previous presentation, the various GEANS pilot studies have shown that DNA-based monitoring can offer an alternative or complementary monitoring method for use in monitoring programs. We also, as shown by Reindert and Magdalini, have delivered important tools and products that can help towards implementation. So in this presentation, I will show you which steps have been taken so far, how you could start implementing, and what next steps are still needed. First, based on the lessons that we learned across all the several pilots, we have formulated a set of overall strengths and weaknesses of DNA-based methods. Since for implementation, we realized that it's really important to have an honest view both on the positive and on the negative aspects, as expectation management really is key. So for the positive aspects, we have shown that, or at least in most cases, they are faster and cheaper, these DNA-based methods. They allow for a higher throughput of samples. Also, the protocols are repeatable by different organizations, and they are robust even if small changes are applied. And this implementation can lead to an increased standardization across regions. We also showed that DNA-based methods are able to pick up community patterns in a similar way as traditional methods do, and they often detect more species, and they're also better in detecting new species, for instance, non-indigenous species, earlier than traditional methods. On the downside then, as shown by Sophie, no quantitative information is provided. They can also be more prone to technical biases. For instance, for certain species, it's more difficult to amplify them. The species lists that we are generating are highly reliant on reference libraries. So this means that the species list is as good as the reference library that you are using. Implementing DNA-based methods might also mean that you have to interrupt a traditional time series, for instance, that's built up in the framework of MSFD. And still, there is sometimes a lack of trust at stakeholder level in these methods. And we see that these negative aspects, that they are hampering routine implementation, since often it's the negative ones that prevail over the many positive ones. As well, inherent to human psychology, people often tend to stick to what they know, which is making actually the road towards implementation quite challenging. So, but nevertheless, we've taken several steps, and let's have a look how this challenging road towards implementation looks like. Of course, it's important to acknowledge that before GEANS, there were already several studies that showed the potential of bulk DNA metabarcoding, and we have been building further on that knowledge. But at the time that the project started, uptake, especially at national stakeholder level, was still very limited. On the one hand, because there was a high ignorance on what the potential of DNA-based methods were, but also there was a level of skepticism, as mentioned, because of the negative aspects. And these methods, they were still very much in a research research stage. So we worked further in uptake in national monitoring programs. This we did by our pilot studies. These were transnational efforts, as you have seen, and they were in close collaboration with national stakeholders and also on monitoring questions that were of interest to these stakeholders. Because it's really important to have the stakeholders with you when you are striving for implementation. And we thought, okay, by showing empirical evidence, that's one way to do so. So we show them that DNA-based methods can be an extra instrument in the monitoring toolkit. So it's not necessary to abandon traditional methods. No, it's actually making use of the strength of both methods. So complementarity is really key here. And this, because that means that when you're integrating both methods, you have actually a more comprehensive assessment, and you can this can actually lead to an increased understanding of what's happening in our marine ecosystem. Another important next step, of course, is reaching consensus on how to do things. So on the methodology, on the protocols, on the workflows. And our pilots, they were leading in this, but as has been told by Reindert and Sophie as well, we also did a ring test. So we came up with a standardized protocol for a bulk DNA metabarcoding of soft sediments, which has been shown to be robust and repeatable, which is important for implementation because harmonization of protocols has the big advantage that it leads to increased standardization of biodiversity data at a regional level. And this is a really big asset for regional assessments, for instance, in the framework of MSFD or other policy drivers. And of course, all the work within GEANS, it resulted in several outputs. As you have seen, these are publicly available on our website, and especially our interactive decision tree. I think it's a good tool to communicate with stakeholders because it guides the user to the most appropriate technology for a specific question. Let's have a look at how it looks like. So Reindert already showed the entrance page. Let's say we are interested in environmental impact assessment. Then you have the choice between different impacts. Say we are interested in impact on soft sediments, and we are interested to monitor the whole community of species. Then we have again several options. The first one is asking, okay, I'm only interested in changes in benthic communities, so general patterns, general trends. Then you come at the final page where we have a recommendation or a suggestion to say, okay, when you're only interested in this, it could be a good idea to use bulk metabarcoding. Of course, what I'm showing here is the recommendation. This recommendation, it's built on the data. It's backed up by data of our pilot studies. And in the tool, you will find links to the pilot reports, to the protocols that we have been using. So it's all nicely integrated there for you to use. If you would say, okay, I'm really also interested in diversity in life stages, in abundance and biomass, then we say, okay, please use a combined approach, combine traditional morphology with bulk metabarcoding. And how could such a combined approach look like? This is just a fictitious example that I'm giving. So there is an impact, and you would be using a gradient design to look at how far the effect of the impact is reaching. In a traditional way, you would take at every distance, for instance, 3 replicates. You would analyze the grab samples in the lab under a stereo microscope, which is quite time-consuming. We would suggest that you combine both traditional and DNA-based methods by taking samples for DNA-based analysis at every distance, and maybe at the locations where you expect the highest impact, also supplement it with the traditional methods that you're used to use. Because this combination, it will lead to a rapid assessment to detect the changes using the DNA-based method. So it will allow you to say, okay, the impact has an effect up to this distance. And by combining both methods, you will also have your most comprehensive species list. And at your crucial locations, you would not lose any quantitative information. So you will have biomass abundance data supplemented by your DNA-based methods. Additionally, this example is now just a small number of sampling locations, but if you would have even more stations, then for a similar kind of budget, when you combine this approach, you would even be able to increase your efforts. So instead of having only 3 replicates, you could go for 4 or 5 replicates. So that would also increase the power of your assessment as another advantage. But going back to our roadmap, so as you've just seen, GEANS has taken several steps on this road to implementation, and actually it paid off. Some countries already started routine implementation. We call them our early adopters, such as, for instance, Sweden, who is using DNA-based methods in their monitoring for NIST. But also, I think, by doing these pilots in the other North Sea countries, awareness on routine application or the possibilities of using it for a routine application has been grown, and first steps towards implementation are really taken. But of course, we have to keep this momentum for continued collaboration, because there are also still some gaps, and we have to close these gaps. So there is also still some work ahead. And which actions do we think are important to take implementation further? The first one is centralized and standardized reliable reference libraries. As you've seen, within GEANS, we have been centralizing for North Sea macrobenthos, but there are other faunal groups as well. So we need to do this for more faunal groups, and also even the one we built can be further elaborated on, and ideally also include other markers in these reference libraries. We also need to come up with even more regional guidelines and methodological standards, especially for eDNA. This is quite an important advancement that we need to make. Also, when considering implementation of DNA-based methods, it's important to, from the start with proper data management according to the FAIR principle. So your data need to be findable, accessible, interoperable, and reusable. And this means having a good provenance tracking. So making sure that it's really clear what exactly has been done with your data, which metadata are related to this. Another important one is that we will need trained people able to do these DNA-based methods, to do the processing of the samples. And within GEANS, communication with stakeholders was key, but this should be continuing, since this really is important towards implementation. And I think it's not an action, but it's something to remember, is that we should definitely not forget all the local taxonomic and ecological knowledge that's already out there from the years of traditional monitoring, because it's good to validate your DNA-based monitoring data. Several of these actions, they are actually followed up in newly started Horizon Europe projects, which you will hear about in a minute. So it will be only one presentation instead of 2, but still. And lastly, of course, I think it's important to keep DNA-based methods and their implementation high on the agenda, lobbied by funding agencies, so we can keep on working towards implementation. Because all these steps in the end will lead to routine application of DNA-based monitoring, and this will result in high resolution biodiversity monitoring of our seas and oceans. And I would like to conclude with making a plea to not wait, but start integrating DNA-based methods in your national monitoring programs, because it will be the fastest way to routine application. We, at the same time, then start with constructing time series in parallel with the traditional one. So then the issue of, okay, we are breaking up a time series is less valid anymore. And of course, when doing so, please make use of our available GEANS outputs. Thank you.